Okay, so from the previous session, they're letting us know that they finished quite on time. Thank you for letting us know. And welcome to this session. I am Kasia Fantonio, iBOL Europe Community Manager, and I am based at Naturalis in the Netherlands. And today, with the support of Pete Hollingsworth from the Royal Botanic Garden in Edinburgh, we are going to host this session about iBOL Europe. So, iBOL Europe is a community of more than 500 members throughout the European region working on DNA barcoding. The iBOL Europe, we work on different areas, so many different research topics, but today we're gonna hear especially on the reference library curation by Brent Emerson and Filipe Costa. Then we will also hear about the ONT and DNA barcoding by Kevin Beentjes. He's also at Naturalis. And finally, we will hear a bit about barcoding museum specimens by Ben Price. So, as I said, iBOL Europe is a network of more than 500 members, and we are kind of the European node of the International Barcode of Life consortium, and we are an open community, so free to and open to new members. And we engage with each other in different ways, including community meetings and webinars, working groups on high priority research topics, and other like newsletters and other engaging initiatives. So I'll soon drop the link to iBOL Europe for anyone in the audience willing to join us. The session is being recorded, and there will be a Q&A session at the end of the session. Sorry for this slip of the tongue. But yeah, so please, you can ask your questions. We hope to receive many in the chat. And yeah, so I guess now I can give the floor to Brent Emerson and Filipe Costa. Brent is a research professor at the Spanish National Research Council, and he is based in the Canary Islands. His research focuses on the structures and spatial patterns of variation within arthropods, from the level of genes within species to species within communities. While Filipe Costa is a marine ecologist and associate professor at the University of Minho, Portugal, where he leads a research group on molecular ecology, biodiversity, and DNA barcoding. So, Brent and Filipe, the floor is yours. Thank you.
Okay. Thank you. Right, can everyone see my presentation okay?
Wonderfully.
Right, thanks. Okay, thanks, Kasia. So barcode sequencing is now emerging as a very powerful tool for the democratization of taxonomy. Okay, by that I mean if every species were to have a barcode reference sequence, identifying the taxonomy of any unknown individual would become a simple sequencing task. However, existing databases contain errors, and these errors can range from simple misspellings through to incorrect taxonomic assignment. And the consequences of this are, well, for barcoding, we can wind up with conflicting or incorrect taxonomic assignment of newly sequenced material, and this can have very big implications when public administrations are involved in conservation and administration of funds. And for meta-barcoding, it can be problematic on two fronts. Again, we can have incorrect taxonomic assignment of OTUs obtained, and also meta-barcoding, we now realize, overestimates the number of species in a sample, but this can be controlled for when reference barcode sequences are available. So just by that, we have, as meta-barcoding becomes rolled out at greater scale globally, we have denoisers that are able to remove PCR and sequencing error, but they're less able to handle co-amplified nucleic copies, and these are problematic in that they inflate variation estimates within species, but they also result in inflation of the number of species themselves. And this inflation can lead to OTUs estimated from numps exceeding the number of real OTUs in a sample. So we can have more than double the number of species being inferred. But barcode reference sequences are one pathway to controlling for this phenomena. So there are very good reasons why we need well-curated reference libraries. And for that reason, within the Biodiversity Genomics Europe project, we have a specific task dedicated to this. So our aim is to curate about 200,000 existing sequences on BOL that relate to 25,000 species to address the current inconsistencies, and to produce and curate 45,000 new sequences from 15,000 species to fill existing gaps. So our main focus is on pollinators, as well as freshwater and marine invertebrates at the European geographic scale. And we aim to produce publicly available reference libraries of European barcode sequences. So our approach is just to summarize. Firstly, we identify inconsistencies in the databases. So these are primarily bin sharing, where two or more species are in the same bin. For anyone who's not familiar with the terminology, a bin is analogous to an OTU. And so this bin sharing can be due to issues of outdated taxonomy, misspellings, misidentification. We can also have multiple bins. So that's when one species occurs in more than one bin or one OTU. And this can be broadly divided into monophyly, which represents cryptic diversity within species, or paraphyly, which again leads us towards issues of misidentification. So the next step is then to correct these inconsistencies and acquire all the available metadata of relevance for a curated reference library. Following this, we then score records from 1 to 6 according to the sequence and metadata quality of those records. And as a function of this, we can then grade species according to their bin sharing and bin splitting. And these grades we use range from grade A, where we have many specimens and all occur in the same bin for a single species, through to grade E, where there are definite issues due to bin sharing, which is more than one species occurring in the same bin. So what I've just described there, we've rolled together into an automated curation pipeline. So all of those steps just described are being fully automated. And this results in a not completely curated database, but a highly curated database that can then be passed to taxonomic experts, typically at the family level, for further manual curation and validation of the pipeline output. And ultimately, this leads to curated reference libraries. Now, in terms of where we're at right now, in terms of progress, we now have our first curated barcode reference library for the geometrids of Europe, which is completed and now being prepared for publication. And this essentially is a proof of concept of our automated and manual curation pipelines. We are now in the process of, so we've also completed automated curation for many other Lepidoptera groups, and the manual curation is now in progress. And between now and the end of the project, we will be focusing on the other pollinators, as well as the freshwater and marine invertebrate species to present their curated reference libraries. I would like to now hand over to Filipe Costa, who is going to present to us from the perspective of the EDN Aqua Plan. Thank you, Filipe.
Thank you very much, Brent. So I will talk about this project, which is a coordination and support action funded by Horizon Europe. And as you can see by the title, it's what we want to do is to develop a plan for a sequence, DNA sequence reference library, and also a data repository that can support monitoring of aquatic organisms in Europe. Next, please. Okay, so this project has 18 partners from 11 countries, which compared to the size of the project and the funding is a good coverage. It's a 36-month project that started in September 2023, includes EMBL and other databases as OBIs, different academia and research institutes, quite diverse. It's coordinated by the European Marine Biological Research Center. And next, please. And these are the structure of the most relevant work packages for this talk that we have. So we have a work package that aims at make an audit and gap analysis of the reference libraries and eDNA repositories in Europe for aquatic organisms. And we use a number of tools like questionnaire surveys. We queried public repositories and also do the large language models to mine papers. And through this survey, we want to then pass on this information on this gap analysis to the WP3, which is about the standards and interoperability of the reference libraries. Then we have a number of use cases in WP4, marine and freshwater use cases from bacteria to vertebrates. And we will implement these standards to these use cases and see if they are feasible, if they are appropriate or not. And then we will feed this information into WP5, where the goal is to make a blueprint, a roadmap, and a sustainability model for a DNA reference sequence library for aquatic organisms in Europe. Next, please. So this is where we're going to focus on the reference libraries only. We're not going to talk about eDNA repositories today. Next, please. So we did already this landscape and gap analysis. We used 40 data attributes that we queried in the reference libraries. And this was concerning all taxa and markers that are used in aquatic organisms monitoring in Europe. We searched for 21 repositories of data. We submitted a questionnaire and we had 38 replies from European countries, European researchers. We acknowledge these contributors. And also we had a large language model that was used to search over 850 publications. It's a long report. It's not fully published yet, but there's already a summary available on our website, so you can check it if you want to have some early information. Next, please. So what were the main gaps and the general findings for this landscape analysis? Overall, the reference libraries have a very high heterogeneity in quality and levels of taxonomic completion. We found that metadata standards many times are absent or are minimal, and there is insufficient or very basal implementation of comprehensive quality assurance and quality control systems. So this is a bit worrying. And also in most cases, there is no post-barcoding assessment of the taxonomic accuracy of and annotation systems. Also, lots of data is not very easily accessible and not interoperable. Next, please. So we feed this information to WP3, and WP3 is now working on building on the literature and our findings in WP2 to develop recommendations for the genetic reference libraries when we want to develop or propose or recommend standards and validation criteria for the reference library records. So we don't want to reinvent the wheel, but we want to use the available information and then try to harmonize with among different initiatives these standards. Also evaluate the systems for curating reference libraries and essential metadata, and also to propose solutions for improving interoperability of these databases. Next, please. Okay, so what we have done so far in WP3 is that we are consulting the community, the experts in the community, and we have been having discussions with related projects and initiatives, including iBOL Europe, looking at what are people doing regarding essential metadata and curation procedures. Also, next, please. We have done recently in last September a workshop where the whole Eden Aquapan Consortium participated, also our external advisors. And we were, during this workshop, discussing storage of vouchers, data and metadata, what are the taxonomic curation procedures that exist and what we should do in the future, and also looking at machine-readable workflows and interoperability. Next, please. So overall, our conclusions is that yes, we still need a strong investment in reference libraries. They are still not complete at all, far from complete, and there's quite a bit of improvement that needs to be done regarding the quality of the data. So there should be still funding for this, and the community should strive to complete these reference libraries. Next, please. And we have a lot of work in progress in order to produce recommendations for these reference libraries. And we are aiming at reference libraries that are accurate and integrates full-scale taxonomic curation procedures, auditable and reliable. Here is very relevant to the metadata quality and standards, harmonized and interoperable with other systems, with the emphasis on FAIR principles, and also that is a sustainable DNA reference library for aquatic organisms. Of course, we have a lot of work ahead, and we will do this in partnership with iBOL Europe and other relevant initiatives, so that we have a broadly applied system that is shared among the community working on these topics. Next, please. Thank you very much for your attention. Thank you, iBOL Europe, for the invitation for this talk. Also, thanks for our external advisors and for the European Union for the funding. And you can see more information in Eden Aquaplan. Thank you very much.
Super, thank you so much, Brent and Filipe. It was extremely insightful to hear your double presentation on reference libraries. I think now we can give the floor to Kevin Beentjes as he walks us through the ONT and the DNA barcoding part. Kevin is the team lead and product owner of the DNA sequencing team at the Naturalis Biodiversity Center in the Netherlands. His work focuses on large-scale DNA research infrastructures to create a complete DNA reference library of all Dutch multicellular life. So please, Kevin, the floor is yours.
Yeah, I had to unmute, sorry.
Yes, please, very good.
Okay. Well, thank you very much for having me. Well, as Kasia already said, I'll walk you through some of the work we've been doing on the DNA barcoding, especially with Oxford Nanopore, mainly how to scale up DNA barcoding efforts. So all of this work has been done within the umbrella of the ARISE program, which is our program that works on a national research infrastructure to mainly do species recognition, not just with DNA, but also with sound, image, etc. But I work mainly on the DNA part, so I'll focus on that today. So when we talk about DNA barcoding, I think Brent already mentioned some of the benefits of having a good barcode library. But of course, that barcode library needs to be built. So basically what we've been doing at Naturalis is looking at this whole process anew to see where we can basically upgrade and change the workflow to scale this whole process up. So we're currently working on a method in which we can process about 100,000 samples a year. And that's with a relatively small team of only 3 technicians in the lab. And our main aim was to lower the cost of this barcode or barcoding of specimens to preferably under €2 per sample. But this means we don't want to compromise on the quality of the DNA work. And of course, bring in a lot of automation to process large numbers of samples. So basically, I'll walk you through a bunch of steps that we changed. Hopefully you get some inspiration for your own barcoding programs. But of course, barcoding starts with collecting material. So what we've done is we created a bunch of standardized kits, which contain everything from tubes, registration numbers, instructions, etc., that people can take with them into the field and then collect material in a standardized way. So we now have these for insects, plants, and fungi. And this is linked to a mobile app, which we use for people to input their data so that everything is nicely standardized and complete before specimens end up in our lab. Then, of course, we want to keep track of what's being done because we don't want to sequence the same fly 100 times. So what we did is we set up what we call a target list. So basically, we took the whole Dutch species list, and we are basically checking off all the specimens that we get in and for which we have specimens and barcodes, so that people in the field, especially these amateur taxonomists that do a lot of work for us, can see which species in their group are still missing from the reference list. Then once the specimens come into the lab, we've basically decided we don't want to rely on commercial DNA extraction kits, but we also don't want to do the really cheap and dirty hotshot DNA extractions, because our DNA extracts will be stored for longer and for later research. We might also want to do some genome sequencing on this, so we need proper DNA extracts. So we basically went to the drawing board and we came up with a combination of two protocols, which is the MuDNA protocol and the BOMB protocol. We did some tweaks and changes, and we basically automated all of this on a big liquid handling station, where we managed to get the cost of the DNA extraction down from €4 to about 70 cents. And we're now at a point where basically more cost efficiency can only be done when we reduce the amount of plastic. So we're also looking at reusing plastics. But like I said, these DNA extracts, the quality and the yield is on par with commercial kits. So they are really nice and clean, and we can do actual genome sequencing, which we have done already for a couple of these extracts. And for the sequencing, we switched from Sanger, obviously, to nanopores. That was the title of this slideshow, where we basically are now in a process of sequencing 24 plates, so these 96 well plates, on a single flow cell, which again decreased the costs of the sequencing itself from about €5 to less than €1.50, which includes the amplification and the sequencing cost. And so basically also what we're doing is now working towards a one-step PCR where the index is already on the primers. So a lot of people still probably use the two-step PCR index. It's a bit of an investment because of course you need to buy all these index primers, but it saves a lot of work in the lab and thereby also a lot of costs. We're looking at could we actually lower the price even more by putting more plates on a single flow cell. But as you can see in this graph, we're now about at the point where just putting more samples on a flow cell doesn't really decrease the price per sample anymore. At this point, because we put so many samples on a single flow cell, the cost of the flow cell itself becomes negligible. Plus increasing the number of samples on a flow cell here only makes sure that you have less data per sample on average. For those of you who don't know, nanopore sequencing, I'm not going to explain the whole theory. There's plenty of information to find online, but basically it's a new way of sequencing where it's sort of a next-gen sequencing technique. So you won't get one sequence per specimen, you get a whole bunch of sequences, which is then nice. I'll come back to that. So for the analysis, the nanopore sequencing comes with a set of standard tools, but we don't use all of those because we do this little trick with the double indexing. So we have some custom scripts we are all now in the process of also publishing. So if you hang on for just a little while, you can just copy whatever we did without having to reinvent the wheel. All the sequence data that we generate is collected in what we at Naturalis call our core sequencing cloud, which brings together all the data from both the specimens that were entered in that app you saw earlier, but also all the processes that happen in the limbs, and of course the sequence information itself. And so all of this information about primer usage, all the details about sequence, etc., are all collected in one spot. So that makes it easier for us to collate this data and ship it out to Bold or wherever. Just for the fun of it, I have some statistics of a couple runs we did. So this is statistics for about 20,000 samples, which generated almost 30,000 barcodes. And like I said earlier, a lot of specimens will not yield one single sequence, but actually multiple sequences, which include some of these nums that were already mentioned earlier, but also what we sequence some parasites and some contaminants, obviously. So there's some usual suspects like the human contaminant, but also the bacterial contaminants that we filter out. Every now and then you run into some weird things like eels or this antelope from Mongolia. Turns out, even if you use Bold, there are still some errors in there of people that uploaded their own DNA or a bacterial contaminant as one of these creatures. So whenever you blast against it, you find these suddenly appearing everywhere, which also under sort of um, um, yeah, shows why this curation of this data is so necessary. We actually had one specimen that gave us 27 different barcodes, but on average, we see that roughly 30% of the samples give us more than one barcode. Of course, then once we have the barcodes, we need to do some validation. So we have a whole sort of pipeline where we check both technical issues with the sequence, but also whether or not it's contaminants and whether or not the a quick blast gives some identification that is at least close to what the taxonomist said it was. Obviously, we're working with a lot of specimens for which there is no reference, so we're not relying on these 100% matches anyway. And like I said, everything comes together in the core sequence cloud, which is a data lake house. You can find more on this on the website, which is linked in the end. And we're now working on a dashboard where people can actually see the status of the samples they send in, and then download their sequences and see what's going on without having to email us constantly on updates for their specimens. Of course, like I said, this is also meant as an inspiration for some of you. ONT barcoding is relatively cheap, so it's not something you need to spend a lot of money to get started with in your own labs. Currently, these MinION sequencers, which fit a flow cell, are less than 2,000 euros. I mean, if it's from your personal budget, it sounds like a lot, but if you compare it to any other lab equipment, it's relatively cheap. And ONT community online has many different protocols, scripts also, but also an active user community. So if you have any issues that you run into, you can just go onto the forum, post whatever it is. There's a lot of people there to try and help you out. And then, of course, there's also opportunities for training. So BGE is doing some training online and in person. There's a link. You can use the QR code here to scan. And of course, different seminars also via the iBOL Europe node. Recently, there was a webinar on DNA barcoding with Nanopore, actually, and there's probably going to be more of these. like I said, we're working on a publication for the whole end-to-end. So everything from the sample kit to the scripts you need to clean your data. That is a work in progress. So the publication will hopefully be there somewhere early next year. But for now, thank you for your attention and thank you for having me.
Kevin, thank you very much for sharing these impressive results and methods. I would like to remind, first of all, to all our participants that you can ask any question through the chat. So please ask your questions to the chat. And now I would like to introduce you to the last speakers of this session. Ben Price is going to walk us through barcoding museum specimens. Ben is a principal curator at the Natural History Museum in London. He is responsible for part of the museum insect collection, and his research focuses on scaling up biodiversity discovery using AI and DNA-based methods. Ben, please join us on the stage.
Hi, Kasia. Thank you. I'm also I'm very good at making very long sentences in my bios, so apologies. Right, let me just share my slides. Right. Can you confirm you can see that?
Yes.
Brilliant. Okay. Right. So thank you very much. So yeah, as Kasia said, I'm going to be talking about barcoding and museum specimens, or what I like to call skimming at scale, and bringing historic collections into the genomic era. Now, why museums and why now? So I'll handle that in two parts. Museums, I work at a museum, so they're wonderful places to visit and to work at. But they really allow us to sort of travel through time. It's the only form of time travel that I know about, where you can go into a collection and go back to species that don't exist now because they've become extinct, to parts of the world that have changed dramatically, completely through urbanization and things like that. And so they offer us this window into the past. And so they have this collection of amazing, unique objects. And so bringing genetics and genomics to them is a fantastic opportunity. They obviously have a very taxonomic complete collection, globally speaking. The vast majority of all described life has been vouchered somewhere in a museum. And they hold type specimens, which are the sort of underpin taxonomy throughout the underpin taxonomy, and specifically in places that cryptic species complexes, being able to actually get genomic data from these type specimens really helps to unpick complex issues. And then one thing that's kind of not really thought about much is that they are frontiers of discovery, in that a paper from almost a decade ago now, I think, showed that half of the new plant species were discovered within collections existing. And so they're frontiers of discovery for new species. So that's museums. But in terms of timing, why now? Well, we're at a point now where technology, in terms of like automation, robotics, and the cost per base for genome sequencing has made it feasible to tackle these things at scale. And you can see that famous graph of genome costs plummeting. But the second thing, I think, is that if you look at this graph that I took off the BOLD website, you can see that there are almost over 1 million bins have been registered in BOLD. And I think the prediction is that by the late 2029 or early 2030s, at least, there'll be more bins registered on BOLD than there will be named species. And so we really have this opportunity to use museum collections in particular to embed bins within this Linnaean framework, so that we can have the DNA-based identification sort of fit within a taxonomic framework that exists currently. And so they can all play nicely together. So museum challenges, there's lots of challenges. The biggest, I think, would be the variety and sample. When you're trying to scale things up, standardization is key to getting a lot of things moving through the lab quickly. But unfortunately, museum samples are, well, fortunately, are quite varied. And you can have wonderful, beautiful 200-year-old butterfly type specimens that look like that example there. And then likewise, you can have chironomids, tiny midges on the head of a pin, and that's a type specimen of a type that we sequenced recently. And they have very little DNA, and two reasons for that, fragmentation and contamination. So a lot of the DNA that we end up sequencing happens to be human or fungal, or weirdly a parasitoid that was living within the insect somehow gets sequenced preferentially. And we also use as little of the specimens as possible, because we want, with museum material, you're always balancing current use against protecting them for future use. And so there's this big question about whether or not we're using the best methods that are available and whether or not these methods will change over time. So DNA extraction in particular, the methods have been quite stable over time. And so we know that the methods we're using are basically the best they probably will be. We also don't have an accurate preservation history for many of our samples. You don't know if they've been in formalin, or if they've been, what percentage ethanol they've been in, who's done what to them basically over the last couple of 100 years. And we have this complex Nagoya protocol sort of access and benefit sharing landscape to navigate as well. And many people think that this just impacts fresh material, things that are collected sort of post 2014, but that's actually untrue. And many countries have national and ABS legislation that goes back in time. And so this is something we have to look into. The benefits obviously are huge of using museum collections. And here are just a really small subset of examples where I've been fortunate enough to work in the groups that have been publishing this. So obviously the reference library gap filling, Biodiversity Genomics Europe project is aiming to sequence over 15,000 species, and 2/3 of those will come from museum collections. Those will fill gaps for pollinators across Europe and really make it much easier for meta-barcoding and things like that, and accelerated taxonomy across European pollinators, and as well as freshwater and marine priority species. But they're also really cool at resolving tiny type specimens, as it were. Here on the top, chironomid midge, again, on the head of a pin. We were successfully able to sequence that one's COX-1 barcode, and I think most of its mitochondrial genome, and show that we could place it into a phylogenetic framework and identify which of these several bins and clusters on BOLD actually are that specimen and that species, as it were. We've also found new species in our collections where, weirdly, an Australian micro moth, very small moth, bottom right there, the species was found in the UK, and we were able to go back to the collections and look at Australian exemplars from about 200 years ago almost, and sequence those successfully, and then describe the new species in Australia based on specimens from Australia originally and also from the UK. And then quite excitingly, this paper has just been submitted by my PhD student, Jack, looking at integrating AI with genomics to help with museum curation. So we've digitized many of our British and Irish moths and butterflies, I think hundreds and hundreds of thousands of these. And the idea was to use AI to try and spot whether or not there were any mistakenly identified things in the collection. And this involved the AI going through the human predictions, and then also having these experts in particular pore over these drawers and try and figure out whether or not it was misidentified or not. And many times, most of the time, the AI was correct, actually, that this was misidentified. However, in some cases, no one was quite sure, so we took those samples aside for genome skimming. Weirdly, in some cases, the AI was right, and in some other cases, the expert was right. So in terms of how do we get DNA out of these samples, there's three major approaches, and I'll go through them relatively briefly. You have the multiple PCR targeting short fragments approach that was published by Sean Prosser and Jacopo from CBG in Canada. And you can see that it has a varied success rate here, costs around about 25 euros per sample, including sort of the lab staff time, but recovers a single gene. And it uses a lot of PCR and a lot of PCR cycles to do that, and doesn't work very well the further back in time you go. Genome skimming, which is the bit I'll focus on here, is essentially whole genome sequencing, but just at a very low coverage. So this costs about 15 to 30 euros, depending on the genome size in particular. These days, we're having about 70% successful barcode recovery from our methods. We sequence all the DNA, the good and the bad, the contaminants and the target sequences. And the idea is that low coverage sequencing allows you to recover the high copy nuclear and organelle genes. And you can always revisit the sequencing library in time if you need more data. Target enrichment is essentially equivalent to genome skimming. However, there's an extra step in that after you prepare your libraries, you can enrich for specific targets that you're interested in sequencing. So that is cheaper to sequence because you're sequencing less to sequence those targets, but you need to develop capture probes, which aren't free, obviously. And you get better target recovery, but it's not representative of the genome. So the current approach is to use the genome skimming approach, and to upscale it, really the drive is reduce reagents and increase throughput. Tissue sampling, I'm going to go through it step by step here. Tissue sampling, the idea is to use minimal tissue as possible, again, to balance against future use. So for example, in pinned insects, we're typically using a leg or an abdomen if we're going to do a genitalia preparation afterwards. It's very hard or impossible at this point to automate tissue sampling. So we have dedicated people working in the labs. They work to prevent cross-contamination by bleaching everything and heat treating everything and working in dedicated spaces. Then we move on to extraction. And once things are plated up into 96 well plates, it's a lot easier to go quite quick. Here's a kingfisher apex, which we've got a modified ancient DNA extraction method on. And I've put some of these QR codes throughout the talk for people to look at. The take home is it's about 70p a sample. Skipping on to library building, this is where robotics help, and there's two approaches that we're using here at Natural History Museum and at Naturalis as part of the BGE project, and that's really reduced volume of commercial kits, so taking a commercial kit and putting it right down to like 1/8 of the volume, so 1/8 of the price per sample, and then an alternative approach that we've done is to use DIY forensic approaches, so the Santa Cruz reaction where you have to create your own buffers and sequencing reagents. And the cost is about 8 to 12 euros per sample. Then we have to sequence them massively multiplexingly. So we use this dual indexing approach. And at the moment, we're sequencing about 380 samples per NovaSeq, which is one of the biggest sequencing machines out there. That costs around 8 euros per sample. In terms of the bioinformatics, I'm happy that the one up at the top right there was published today. We got the notification. We're using a couple of approaches. One is to target single genes, so mitogene extractor, just to target the COX-1 barcode. Second approach is using skim to mito, which is assembling the organelle genome, and then we can chop out the genes of interest that way. And then we throw them all through Barcode Validator, which is a little GitHub bot that checks taxonomy and various other metrics. And the key here for upscaling is really the target reference sequences. And so we've got some scripts that we're working on at the moment to sort of automate getting the reference data that you would assemble against. So it's just winding this up a bit. So some lessons learned. Well, yeah, the sequences do strike back. It's very difficult to predict success with museum samples because of the varied input. Something that looks beautiful and it was collected 20 years ago might sequence very poorly, and something that looks horrible and was collected 150 years ago might give you a whole mitochondrial genome. Use dedicated facilities if possible. Robotic implementation takes quite a bit of time, and we don't always need robots. One of the lessons we've had is that just buying magnet stampers works really quickly. The quick wins to reduce costs are really around volume of kits and DIY reagents. And right now, the current cost is around about 15 to 20 euros per sample for insects at least. And that excludes staff. And with our two-person team, we've got about 8 plates per month. And that adds around about 20 euros per sample again. So and the only way to decrease that is really high throughput. We have to increase the throughput in the labs. And so looking ahead, we're refining our protocols. We're aiming to reduce our waste. We're increasing our throughput by processing many more plates in parallel or moving to 384 well plates. And we're developing our assembly and mapping pipelines because we find that even with our current pipelines, there's a huge amount of variety and no single best method for a sample. And yeah, what will these millions and millions of sequences or samples at least that are in museums around the world, what will they tell them? Will tell us what are the secrets that they're going to release, and we're going to explore the rest of that genomic data over time. And just to close by thanking a lot of these people and especially all the curators and BGE project tissue sample providers. Thank you very much.
Thank you so much, Ben, and congratulations on the fresh publication.
Yes, thank you.
So I am happy to announce that we officially entered the Q&A part of this session. And we already have a question to Kevin from Filipe. Kevin, have you tried it with non-insect invertebrates? Sorry, I got lost. Have you tried it with non-insect invertebrates, and would there be additional adjustments regarding DNA extractions and primers to be used, assuming you are using only one primer with insects?
Oh yeah, I can say we have. So of course the majority of what we do is insects, but that's the majority of the fauna. But we have indeed also processed a lot of marine invertebrates. DNA extraction protocol works quite well without any adjustments. For PCR, we are using some different primers than we do for the insect. But the changes there are not very different from what you normally do. So you do probably need to get some inhibitor removals or dilute your DNA a little before you do your PCR, but it's relatively comparable to what I hope, Filipe, you are already familiar with. So, yeah.
Great, thank you, Kevin. And we have straight after another question for you, very popular. So, is your sequencing barcoding workflow challenged by the nanopore error rate? And this is by Maria Zolotareva. Apologize for the pronunciation.
No, because we are aware, of course, that Nanopore has a slightly higher error rate. But what we do is because you generate a lot of reads per specimen, we built a consensus sequence out of at least 30 reads now. So that sort of removes all the errors, and we have checked and compared to Sanger data, and it's basically the same sequences. So making sure that you are not using the individual reads, but really building this consensus from the reads that you get negates all these error rates. Yep.
Pete, please, you join the floor.
Yeah, so it's a question for Filipe and Brent about reference library curation, and it's one of the fascinating things about it is it's easy to say let's have a curated reference library, and then you kind of immediately realize what does a curated reference library actually mean? And I just kind of wondered what your feeling was about the one level you can make a kind of curated reference library and say that's it and kind of lock it down and use it. And of course there's new data coming in all the time. Do you have a kind of sense for how dynamic these curated reference libraries need to be, and kind of managing this challenge of getting something all nicely cleaned up and then a whole load of new data comes along, some of it good, some of it bad? Apologies for the diffuse nature of the question, but you get the general gist.
Oh, yeah. Do you want to answer that first, Filipe, or do you want me to have a go?
Go ahead, and then I'll complete.
Okay, yeah. So I think it needs to be dynamic for sure, because within our whatever reference library we're curating, there will be species with very few records that don't fall within the reference library for that very reason. And as we accumulate data, they can eventually reach a grading that allows them to enter into a library. So yeah, how we maintain that is probably the harder question, like over a long time. But some continuity and processing, or at least processing new data, should happen, because we're going to be generating a lot of data over the next 10 years. I mean, a lot. So that will need to be curated. Yeah.
Yeah, I agree with Brent. It's absolutely, you need to update it regularly. I mean, taxonomy is inherently a dynamic process, and we would lose a lot of information if we keep some reference library just stable without regular updates. And what we also see sometimes is that even those records that appear to be very solid and accurate as new data is gathered, sometimes they become, we see some uncertainty in those records. And there's also the lots of situations where we have hidden diversity within a known species, and we can only do that with updated reference library, curated reference libraries.
Great, thank you. I see on the chat there is a question for Ben by Aidana Bekiaitieva. Apologize again. Dear Ben, do you have any modified protocols for DNA extractions from formalin preserved historical specimens? Thank you.
Well, if I had money for every time someone asked me about formalin, yeah, formalin is a bit of a tight one. So we do, and we can share those protocols more widely. I think colleagues at Naturalis have done quite a bit of work on formalin preserved material. The key seems to be using a boiling step to break the crosslinks that formalin produces within DNA. But yeah, I'm sure there will be a document sharing via the iBOL Europe website would probably be the best place for these sorts of protocols.
Yeah, good hint. Thank you. Okay.
Can I put orally a question to Ben? Ben, so this study you have just published using AI for curation of the reference records. What is your feeling about it? Would you envisage AI being a long-term solution for curation of reference libraries?
So in this case, you're talking about the AI of the museum specimen reference library, right? Yeah. Okay. Yep. So I think so. Yeah. I think the benefit of AI is that you can train it on a collection and then make the tool available to other people. So I mean, we were quite shocked at the number of mistakes that we found in what is a fully digitized and curated collection, you know. And I mean, there weren't thousands and thousands of mistakes, but there were still enough mistakes that the AI had to be trained in a sort of multiple iterative approach, because if you include the mislabeled specimens within the training, then you're training the AI to identify something incorrectly, basically. And so we found that there were, Jack developed this technique that sort of did this iterative sampling from the reference specimens so that you reduce the chance that a mislabeled specimen would actually be part of the training, which obviously would completely circularize the whole thing. But I think, yeah, I mean, the short answer is, yeah, I think these sorts of tools, once more and more collections are digitized, will just be invaluable to help curate. Yeah. We also need the experts to verify them, though. So we can't just leave AI to do everything.
Great. Thank you, everyone. So as there were many references throughout this presentation about BGE, and indeed the next session after this one at the BG24 will be around this BGE, Biodiversity Genomics Europe, which is an EU-funded project, which many of us are part of. So I would suggest that we close our session here and reconnect in the next session around BGE. I would like to thank all of you, the speakers, of course, for their excellent presentations, and all the attendees for joining this session. We hope to see you soon within the iBOL Europe community. And well, thank you again. Thanks, everyone.